Okay, so now we have begun mitosis. So the DNA is replicated in interphase before mitosis even occurs. And so this is an overview right here of mitosis. It has 4 different stages. We have prophase, we have metaphase, we have anaphase, and we have telophase or telophase. You can say it either way. So a way to easily remember these different stages is the acronym PMAT. So if you want to go ahead and write that down. So we're going to go ahead and talk about our first stage, which is called prophase. So in prophase, we have chromatin condensing into chromosomes. So right now, you have your model showing interphase, and you have all of those pieces of DNA are all mixed together, kind of like a bowl of spaghetti. So they're all mixed together, but if you were to separate them out, we could have problems with getting knots or tangles, and it wouldn't be the most efficient way to spread the DNA into 2 different cells. So what has to happen first is that the DNA has to condense. So we're going to actually replace those 2 strands of blue yarn and 2 strands of white yarn with instead our pipe cleaner. So it's going to condense into a form that's smaller and more compact, like our pipe cleaner. And if you notice, we have 2 pieces of blue pipe cleaner and 2 pieces of white pipe cleaner. So when we have these, they're going to come together. So we have 1 piece of blue and another piece of blue. So they're coming together to make an X shape. So this is forming our chromosomes that are condensed. So it makes an X-shaped structure, and each of these X's each half of the X is called a sister chromatid. So if we go back here, if we look at our pipe cleaner, each individual pipe cleaner by itself is a sister chromatid, and then they come together. So this blue guy is made of 2 sister chromatids. As well as this white chromosome is also made of 2 sister chromatids. And each of these are attached in the center, and that where it attaches in the center is called the centromere. So if we go back here, the centromere is right here, where the 2 chromatids are attaching. So some other things that are happening in prophase. Prophase is probably the most complicated step because it's got so many things going on. So we're preparing everything, getting everything ready for mitosis to occur. So another things that are happening are that the nuclear membrane is disappearing. So you can see that in this picture up here where the nuclear membrane is starting to break down. That's because if we're going to split the DNA and have some go one way and some go the other way, it can't be confined to that membrane of the nucleus anymore. Oops, moving everything around. So that's going to happen. We also have centrioles and microtubules that are going to appear. So that's shown up here at the top where we have centrioles and microtubules. So those are the machinery that are going to attach onto the chromosomes and physically pull them apart into 2 separate directions. So those are the machinery that's going to do that. And we have spindle fibers that are going to attach to the sister chromatids. It's not really shown a whole lot in this image, but they're going to attach to the sister chromatids so they can actually be pulling the 2 chromosomes to different directions. So right now in prophase, your cell should look like this, where we have our 2 chromosomes, each one made of 2 sister chromatids in our cell.